I'd like to introduce Dr. Mike Zaragoza and Anya Grosberg and have you welcome them for our evening lecture.
Thank you very much for the introduction, and thank you for coming here at the end of, I'm sure, a long day. We are going to try something that we don't normally do in a scientific talk. So we want to have a conversation, and we're going to do this as a conversation between the two of us, and hopefully between us and you as the audience. So please bear with us if we get confused with any of the slides as we try to do this new thing. So we're going to be talking about the heart, and we're going to be talking about heart disease. So in this case, all of you probably know someone who has heart disease. So how many?
By a show of hands, please raise your hand if you know or knew someone with heart disease. Okay.
So we see a lot of raised hands here, and that is not by accident.
So unfortunately, as you know, heart disease is deadly, and the mortality or death rate is quite high. So if you look at this particular slide, it lists the top 10 leading causes of death in the United States. And by these bars, it shows, it shows the relative number of people who die from each of these causes. So you could see very well, number one is heart disease. Heart disease, there's many different types of heart disease, and you could see how comparative it is to cancer, other causes of death in the United States, accidents, lung disease, stroke, Alzheimer's disease, diabetes. But by far, you could see the top two leading causes of death in the United States is heart disease and cancer. And in fact, 1 in 4 of every death in the United States is due to heart disease. And this is true in California. This is true for both males and females. The risk of dying from heart disease, unfortunately, increases as you age. So it's a very common problem.
So when we talk about the heart, what are we actually talking about? So this is the organ that is beating in every single one of you right now. So I'm very, very sure that every single one of you has a beating heart. So this is what the heart looks like in a drawing presentation. This is an MRI image. This is actually my heart when I was in graduate school because one of my mates was doing a study. So you can see the blood flowing through this heart. This is more of an artistic representation of the same thing, and we need this blood to flow. This is what delivers oxygen to every single tissue in your body. And you can also see from these movies that the heart is a very dynamic organ. So this is open heart surgery at the bottom here, and you can see the heart twisting. And in order to produce this motion, in order to pump the blood, the heart needs to produce, the heart muscle needs to produce force. So the main thing that the heart needs to do is to be able to produce force. And then if you think about it, the heart is absolutely amazing. It's a pump. It works for almost 100 years, and it beats almost once a second, right? Or if you're running, it could be even faster, right? So the fact that it works is absolutely amazing. And yes, sometimes things go wrong. So there are many different types of heart disease. And this is an example of a structural heart disease. So what you see here is a normal heart on your left and an abnormal heart. So this is a dilated heart. So you can think of it as a normal heart has a shape of an American football, while a dilated heart has a shape of a basketball. And you see other problems where the walls change thickness, and this morphology change, this shape change, makes this organ as a pump less efficient. So the heart on the left can pump significantly more blood out than the heart on the right. But this is not the only way it can change. You can have the reverse change. So this, again, the very left image is a cross-section of a normal heart, and then this is hypertrophy. So here the walls are actually thickening, and then the chamber becomes small, and again, you can't supply this oxygenated blood to the rest of your body, and as a result, you're going to be tired, you're not going to be going up the stairs as easily, etc. And at some point, you might die. So there are other heart diseases as well.
And well, you ask, well, how does the heart control the pumping? You know, because as we're sitting here, as Anya said, everyone's heart's pumping, but it's going at a certain rate and rhythm. You know, so the heart has very specialized parts of it. And these parts are in the boxes, the SA node and the AV node. So these are the parts, are what we call the pacemaker of the heart. So what it does, it controls the electrical activity, the impulse that passes from the top of the heart to the bottom of the heart. And when that happens, it controls the contraction. So therefore, as you're sitting here listening to this talk and your heart rate is beating, it needs to be controlled so you have a regular rate, like Anya said, maybe between 60 to 80 beats per minute. If you're an athlete, runner, like Anya, maybe it's like 50 to 40 beats per minute. If you're giving a talk in front of an audience and trying something new, it might be like 80, 90 beats per minute. So this is the rate, so it doesn't go too fast. And it also has a nice rhythm. Ba-bum, 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 ba-bum. So it's got to be nicely controlled, so therefore the heart muscle can contract regularly to pump that blood in a regular manner. So there's a clinical test, and I'm sure you're aware of this. You might have this in a doctor's office, an ECG, also known as an EKG, electrocardiogram. Has anyone here had an EKG or ECG? Many times for athletes, for students, they would need an ECG from the physician to clear just to make sure that the electrical impulse that goes through the heart during a heartbeat has a regular rate and rhythm. So they put the electrodes on the chest, over the heart. They measure the electrical activity during a heartbeat, and that corresponds to the heart contracting. So during the cycle, when the heart is relaxed, that is in diastole, so muscles relax, it's filling up with blood, and then after it fills up, it contracts. And that's where it pushes the blood from the heart into the rest of the body, also known as systole. So based on the electrical impulses and the EKG, you could see how the heart contracts nice, regular rates and rhythm. Now, there is heart disease also associated with these rhythm abnormalities. So in the upper corner over here is an EKG trace, which is normal. And then I just want you to appreciate the vertical lines and how even they are so that's the heartbeats, a very nice regular rate and rhythm. And all the deflections is similar, okay? It's nicely controlled. However, with heart disease, you have something called arrhythmia. So that's a collection of disorders where your heart is beating too slow, or bradycardia, or it's beating too fast, tachycardia, or as illustrated here on these EKGs, there are parts where the heart beats when it shouldn't. Maybe the beating is starting from the bottom of the heart, the ventricle, and that can be serious. There's a premature beat. Maybe some of the beats are being too close together, uncoordinated, or even the beats coming from the bottom of the heart, the ventricle, and it's way too fast, and then becomes chaotic, that is quivering. That's ventricular fibrillation, one of the most serious forms of arrhythmia, and can lead to sudden cardiac death, okay? So with all this heart disease, what do we do about it?
So maybe some of you can tell us what you might have had a conversation with your doctor, or a conversation with your family. What do you do for for having a healthy life? Because that's part of cardiovascular health. Does anybody? So who's had a conversation. Do you want to start us out, Mike, with something?
Well, yeah, so for your risk factors, certainly what I try to do is watch my diet. Okay, I try. Okay, to watch my weight, because certainly if you're over than your expected weights, then that could contribute, to your risk of heart disease. Okay.
So anybody want to chime in of what you do? Please.
Reduce salt intake.
So reduce
Absolutely.
salt intake. That's absolutely a great way, especially if someone is at risk for having a high blood pressure.
Absolutely.
So anybody else? Yes, please, in the back.
I try to avoid like white bread and rich flours into whole stuff.
Absolutely. So again, diets, plenty of fruits and vegetables, watch your weight.
And as Mike mentioned, if you exercise, so I like running, so any type of, it's actually called doing cardio, and that's because it's really, really helpful for your heart. So there's all of these different things that you can see in this cartoon that are helpful and make your heart happy and make your heart love you. And unfortunately, these things don't work the same for everybody, because you oftentimes hear stories of someone, she exercised, she had a great diet, and then she had a heart attack, or stories like that. So why is it that you could have such a situation of unevenness?
Well, there are many different types of heart disease, many different causes. And as we know, family history can influence your risk for heart disease. So for example, my father at age 45 had high cholesterol, hardening of the arteries, he had a triple bypass, and then he died at the age of 74 a couple of years ago. So family history, there are families in which the risk is elevated.
And you'll find that if you think and you ask your families, there might be history in yours. For example, I know that my grandfather also died of heart disease.
So one thing we can do is look at a family history, see that information, and what we do what we call a pedigree analysis. So a pedigree is a chart that represents essentially a family tree. So we put in different symbols where we could plot the different generations on the left, 1 to 3, and then use different symbols to rep individuals in your family. So squares are males, circles are females, and the lines are relationships. So for example, right below them, when they're connected, they could be siblings. So as you speak with your doctor, geneticist, we can take a family history and see what's people's risk for certain diseases, whether it's inherited or not. And then, perhaps offer genetic testing and, help determine what kind of risk you might have. In these cases where there might be a higher genetic risk than other individuals. Okay. So why is that? So when you think about inheritance, family history, what makes us different? What makes individuals different? What makes, even within your family or different families between ancestries? Where if you look at the hardware, biologically we have cells, 1 trillion of cells. In each of those cells we have chromosomes, and that contains our genetic information. Biochemically it's in DNA, and parts of those DNA contain genes. So think of genes as a recipe or a blueprint, and they encode instructions for proteins. And the proteins, these do the work for the cell. It allows the cell to do a proper function, allows the tissues, the body to make a proper function. So when you look at the hardware, there's a variation, and this is what we call the human genome. So the entire collection of your genes is the human genome, and those genes are packaged into these books that we call chromosomes. So think of chromosomes as books. You look at the pages, those are the DNA, and you can do a test where you're actually looking at the letters on the page to look at the blueprint or recipe to see if there's any abnormalities, any changes to any of these 20,000 protein-encoded genes that make up the human genome. Okay? And then so by combining family history, we could take a family history of individuals, see what kind of health history they have, how old they are, perhaps the causes of death, and then see the relationships, and then we can map out a certain disorder. So for example, if someone has heart disease, we can shade that in, and you could see in this example, there are multiple individuals, both males and females, in multiple generations with heart disease. So when you see families like this is on the other spectrum. I mean, the heart disease is very common, many different causes, but there are some families in which the genetic risk is very high, and often we see that when there's multiple affected individuals. So this gets us to where we're going to focus the rest of the talk, in which we start to study 3 different families with hereditary heart disease. And what we want to start off with is this and this.
We have a strong family history of sick sinus, which leads to cardiomyopathy. We have documented it for 7 generations. My husband and 3 of his siblings have pacemakers. One of his sisters also has a daughter with the same heart disease. Another sister had 2 sons with the disease, but both of them have died already. One of his cousins, age 54, recently died from the same disease, which causes the heart to go from a beat of 32 beats per minute to V-fibrillation. His heart went into V-fib while driving. Those who have the disease have had normal heart function until the disease begins to make itself apparent, usually sometime between 45 and 59 years of age. We are concerned for our children and would like to find someone willing to do a genetic study of the family so that those who are susceptible have a better chance of living a longer life.
The study began in 2008 when she contacted me and told me about her family history. And you can see it's quite striking. So in this first family that we started studying shortly afterwards, we worked with them and drew their family tree. So you can see again the 4 generations over here. Her husband has the history of heart disease. So at that time, he's 68 years old. He had atrial flutter, so the top of the heart began to vibrate and flutter at the age of 54. He had many types of heart disease, VT, ventricular tachycardia, where the lower part was beating too fast. He had sick sinus syndrome, where that pacemaker was diseased, and then it couldn't pace at a proper rate. So the heart became slow, stopped. So they put a pacemaker and an implantable cardiac defibrillator. So defibrillator are like those shock paddles, but this is a little device that's implanted into the heart, measures the rate and rhythm of the heart. Because his heart's sick, it tends to stop. So then he would get a shock. Okay, so a life-saving device, and you could see here, he's right over here, shaded in with the heart disease, the 3 sisters with heart disease over here. One of the sisters had 2 sons who died at a very early age 42 and 35. She had also talked about his cousin at the age of 54, and he was driving and he had ventricular fibrillation. That's when the lower heart began to quiver and he died. He ran into a tree. So she wanted to know what's happening. So a very striking family history here, males and females. 8 people died from the heart disease, cardiomyopathy, as Anya mentioned, the arrhythmia, sudden death, and you could see the ages over here. So very, very striking family history. And then shortly after that, we started studying a second family. They had a very similar cardiac history. Now, the first person that was identified here was a 58-year-old female, and she had syncope, so she lost consciousness for no apparent reason. They found that her heart rate was very slow. So in this case, at age 45, she had a pacemaker to make sure that is beating at a good rate. Later at age 55, she developed the remodeling disease that Anya talked about, the dilated cardiomyopathy. So the heart became floppy and it couldn't pump blood. And at age of 55, just like the other individual in the other family, had one of those intracardiac defibrillators. And then when we took the family history, again here in 4 generations, please just look at the ones who are shaded. Here is the 58-year-old female over here. You could see individuals with cardiomyopathy, arrhythmia, sudden cardiac death at these ages. Shortly afterwards, we met a third family and started to study them. And this family came to our attention. At the time, the first person we identified was about 54 years of age. Several years after he was enrolled in the study, he died. At the age of 29, he developed dilated cardiomyopathy, had a heart transplant at the age of 35. Okay, that's quite young. Now, unfortunately, he had the more common type of heart disease, coronary artery disease. So his family had high cholesterol, and then he had hardening of the coronary arteries, had a heart attack, he had 2 stents placed at age 50 and 53. And then here is his pedigree, and again, here are 4 generations. Defectives are shaded in, males and females. And again, very similar to the other 2 families, striking family history of heart disease, very serious type with cardiomyopathy, arrhythmia, sudden cardiac death. And again, you could see the age is quite young, between 23 and 57. So we started working with these 3 families, consenting them into our study. And as you could see from the introduction where the wife of one of the patients spoke, it was a mystery to these families, okay? There was this family history of people with very severe heart disease, sudden cardiac death, and they didn't really know what was happening. And they wanted to know why. You know, what was causing this? And I find that many of the patients who have, these kind of family histories, aren't really looking at themselves. They're really looking at their descendants because they want to know who their children, for example, for her and her husband, their 5 adult children, and they have, from these children, actually about 12 grandchildren. They want to know who's at risk. Who's going to have the family disease? Who's going to risk of having a pacemaker or heart disease or even die very early? And finally, the families and patients want to know, what are you going to do about it? If we could understand this disease, identify who's at risk, can we actually intervene and prevent? So that's what we started to do. And fortunately, with the tools that we have in genetics, medicine, biology, we're able to look at these 3 families. So we consented these families, we got blood samples, saliva samples, we did DNA sequence analysis to look at the letters, look at those pages to look for the typos, that is, mutations. So after several years after we did that, for each of these family, we found a genetic mutation in a gene called laminase C. So for family A, these are representation of just the data of the sequencing. And we found a certain type of mutation for family A called a splice site, family B a nonsense, and family C a missense. So different types of mutations in the families in the same gene called laminase C. So this was, of course, a high point because we found a genetic mutation for these families, and we were able to develop a genetic test. So now instead of sequencing the entire library or the books or look at all the pages, we had a very specific DNA test, a blood test, that we offered to the family members to see if they had the mutation and would be at risk for heart disease or if they did not. So from these 3 families, over the years that we've been doing this study, we've tested 56 individuals, and then roughly half of them are mutation negative. So they did not have the mutation. So they did not have the risk of heart disease. So this, you can imagine, was such a big, burden lifted off of these individuals, okay? Because in a family situation like this, if you're related to one of the affected, essentially you have a 50% chance of having the disease. So by developing a test, we were able to tell the individuals who they will not have the disease. Now, this is 29 individuals, but you can imagine, depending on where you are in the pedigree, this affected many individuals. So for example, by testing his unaffected sister, and I did show all their descendants, when she tested negative, it cleared everyone below her, all her descendants, okay? So we're talking about not just 29 individuals, but we're able to really give a very, a good news for many parts of the tree, okay, the descendants. So that was wonderful, and that was one of the highlights of this study. Now, what about those other individuals? So of the 56, We also identified 27 which were positive for the mutation, okay? So these individuals, many of these are asymptomatic. They have no cardiac disease, no symptoms, no signs. They wanted to be tested, and we tested them, and they had the mutation, you know? So what could we do for them?
Right, and it's a very tough question. And then one of the things that we start looking at is, what is this mutation? So when we're talking about, oh, there's this lamin AC mutation, what is this coding for? And if you look, so on the bottom here, you have a picture of some cells, and you have, in blue, you have the nuclei. Or you have the schematic at the top on your right of a cell, and inside it shows a nuclei. And the main schematic I want you to look at is the nucleus on its own. So this lamin AC codes for this white net that goes underneath the nucleus envelope. So it's called the nuclear lamina, and one of the proteins in this nuclear lamina is lamin A, and another is lamin C. So why is this important? It's important because this net contributes to the stability of the shape of the nucleus. So this is an example that we have imaged in lab of different nuclei shapes. So at the top, on the very left, you have a normal nucleus. It has very smooth walls, etc. And then you have all of these different defects that you can have. My favorite one is over here because it's a heart. But it is a defective nuclei. So this is potentially caused by a problem with these lamin proteins, but it can also be caused by aging. So as we age, all of us will have more and more nuclei in our tissues that have these defects.
And one thing we ought to mention is this lamin AC gene is also part of a group of disorders called laminopathies. You know, so for our families that we're studying, they particularly have heart disease. However, there is this particular rare disorder called Hutchinson-Gilford progeria syndrome. So this is also due to a mutation in the lamin AC gene. And for this particular one, it's a quite severe pediatric disorder in which individuals, children, when they're born, they look normal and act normal, and then they have very premature, rapid aging. And then they develop many conditions that are associated with aging in the normal population in terms of the hardening of the arteries, atherosclerosis, cardiovascular disease. So these children often die from heart attacks. And then weakening of the bones, osteoporosis, and early death, as I had mentioned, typically about the age of 13. You know, so this particular gene has this severe clinical features and phenotype with this condition, but in our study families, it's restricted mostly for the heart.
And it's actually that this disease almost makes more sense to us because you have a nucleus in many, many cells in your body. So if there's something wrong with the nuclear lamina, then it makes sense that the body overall is affected and you might have this early aging. But when we get biopsies from patients.
Right, so we thought we would start to determine, well, what's the mechanism? Why is this mutation? So we from the clue, we said, well, why don't we get some cells from our patients? So here again, here's the three families, and we started to get skin, because actually the skin in progeria patients are affected, so perhaps that's a good clue to start with. So the patients, and they're boxed in red who are affected with the mutation, boxed in blue, unaffected without the mutation. The family volunteered for skin biopsies, and they gave us skin that we collected that we're able to grow, including the unaffected 15 and all the patients from the three different families over here with the mutation, and we began to study as a first step looking at their skin.
And we're studying the skin because, yeah, you might consent to having a skin biopsy. There are very, very few people who will consent to having a heart biopsy. I will not, and I will bet that most of you will not as well. But unfortunately, when we looked at the skin cells, so this is an example of looking at skin cells from one of the patients, we don't see any striking differences in their skin cells between the patients and the people without a mutation. So you have a collection of these green nuclei that are normal and the red nuclei that we mathematically determine to have these defects. So if you look at their skin cells clinically, they don't have a problem. Mm-hmm. And we don't see it in the lab. So what can we do? We can only get their skin cells, and then we don't see a difference.
So also, fortunately, one of the new tools in biomedical research, as you heard Dr. Anderson said in the beginning, are stem cells. So we took advantage of this new fantastic tool in which we actually develop a model of a disease in a dish. Okay, and then so we developed these stem cells called induced pluripotent stem cells, in which you could take a mature cell, a differentiated cell. So this is a highly specialized cell like a skin cell. And then in the laboratory, reprogram that, as Dr. Anderson said, erase its memory, for example, so that it's a very immature cell or an iPSC cell. And then the second step is to take that, because that's a great tool. Those can self-renew. You almost have an unlimited supply. And then you could differentiate that. That is, take them in the lab and push them into different developmental pathways. And you can make pretty much most tissues, including lung tissue, liver tissue, brain tissues, and we're interested in cardiomyocytes or heart tissue. So we took advantage of this system from the skin biopsies that we have from the patients and unaffected individuals. We took the skin cells. So this is a picture of tissue culture of those skin cells. And if you could see it, I know it's kind of hard to see. These are the skin cells, or opposite fibroblasts. So it's on a dish, two-dimensional growth, and then all these dark squiggly things are the fibroblasts, the skin cells. So you could see there's hundreds of them on this dish growing, and then you could take them to the lab and then reprogram these, so erase the memory. These are thinking, well, we're skin cells, and then bring them to the lab and erase their memory, so they're reprogrammed. They become these big colony of cells, of stem cells, and then you have to isolate them or bring them away from the other cells and then separate them and plate them. So this is, again, another tissue culture dish, the bottom, and then this is a colony of the stem cells, so there are thousands of these cells, and you could see how much different they are from the original skin cells. And this process takes about a month, and you can grow them up and then reprogram. So that's a fantastic tool to take advantage of to model this disease. Now, the second part that we wanted to do was to take all these stem cells and make heart cells. And how do you make a cardiomyocyte? So what we did was looked at nature. In the mouse, in heart development, there's a very early embryo. Now, the induced pluripotent stem cells are very similar to these embryos. They're not derived from embryos, okay? They're not embryonic. Remember, the stem cell that we're working with is derived from the skin, okay? So they're much different. Very similar to this stage, very early embryo, and as the heart develops, it goes into different tissues called the mesoderm, develops into, more specifically, early cardiac progenitors. This part of the embryo develops into parts of the heart, and then very early on, you have this tube as the first structure, and this is when it starts beating. And eventually, and certainly this is more complicated that I'm making this out, it loops and becomes the adult heart, a very complicated structure. So now we can use this, what we know from nature, to...
Wrong way.
I'm going the wrong way, aren't I? Thank you. To mimic this, okay? So we could take our iPSC cells in the dish, grow them up in the well over here, and then pretty much replicate what's happening in nature, where we activate and inhibit using different chemicals to push them into these different developmental pathways. And by day 8, roughly in the dish, you could see over here a sheet of beating cardiomyocytes.
So the problem with this sheet of cardiomyocytes is that it's actually pretty disorganized. And if you look at a nice adult cardiac tissue, the pink image at the bottom on your right, you'll find that it's nicely organized. So this is a very laminar sheet, and if you look at an individual cell, which you have an adult cell at the top, it's nice and elongated, and you see those striations, and the fact that the striations are all aligned really, really matters for how the cardiac muscle produces force. But if we just throw a cardiac cell down on a dish, like you saw in a previous movie, what you actually have is on the bottom on the left, and it's disorganized, and the striations are every which way, and you can think of if everybody's pulling in different directions, you're not going anywhere. So we need a pathway to actually organize this, and what we use is something that most of you probably played with as kids. So who had stamps as a child? Stamps? So those of you who haven't maybe had your passport stamped, although they went to digital now, so it's not as fun of an analogy. But we're using basically the same technology, we just scale it way, way down. So we make the stamps to have whatever pattern we want, and then we ink it with the glue that the cells adhere to. So this is an example of no pattern where we just threw in this glue. And here we pattern these lines. And then as a result, we can create what you see on your right at the bottom, these anisotropic tissues that look pretty much like a section of an actual heart. So we can do this with actual patient cells. So this is an image from cells of an actual patient that was made from skeleton cells into heart cells and then engineered into a tissue. And then we can take this, and we have to ask ourselves, we actually want to compare it to what's happening with the heart. We want to compare it to, we want to know if this thing is capable of producing the same forces. Does it have the same electrical activity? So we ask, can we do that? And what we do is we have another engineered tool. So we take these sheets that we micro-pattern, we stamp, and we put in this layer of a sacrificial polymer. Think of sugar that's dissolving. It's a little more complex than that, but similar. And we put it only on part of it so that you get this flap coming up. So as the cells are contracting, you actually have this thing curling, and it will curl more and more the stronger the cells are. And you see this flap, the movie playing is the flap is coming out of a screen towards you. And what's really cool about this technology is we can put many flaps together onto what we now call a heart on a chip. And not only can we put these flaps on there, we can also lower down a pacer. So this would be like a defibrillator, like the pacers that Mike mentioned, and we can drive the contraction to happen at the same time, very similarly to a way it's driven in an actual heart. And doing a bunch of programming, so I know there's a lot of high school students, so if you have a programming class, it's actually relevant to biomedical engineering. You can track all of these automatically, and you can extract what the force is as a function of time. So what's really cool about this is we can actually compare now these engineered tissues to, it's really hard to see here, but there's on the left of these schematics, there's a red line, that's actually a muscle strip. So somebody took a heart out, cut out a strip of a muscle, and measured the amount of force. So you can think of it being inside a force transducer, you can measure the amount of force it outputs. And these gray boxes are what they measured. And the red and green bar are the systole and the diastole that Mike mentioned earlier. And we show the same amount of force, the same amount of maximum and minimum force that our tissues can produce compared to what would happen if you take an actual heart and take the cells out and look at the actual heart muscle. So we're building something that is equivalent in terms of the amount of force So this shows that if we now build something from stem cell-derived cardiomyocytes, it tells us something about the heart. And we did that, and this is an actual patient, and you can probably immediately see that they're not doing as well. And these red bars are the amount of maximum force they're putting out for the patients, and the blue is one of the people without a mutation, and you can clearly see that the blue is much higher. So there's also, if you look at the details, you'll notice that these films are not going together. That's because it looks like there's also an electrophysiological problem with these patient cells. So what we have created here is a platform that allows us to identify what is functionally wrong with the heart cells of these patients without actually having access to the heart cells of the patients. So this whole platform, the idea is that we replace the need to actually get access to the hearts of the patients, and we can look at structure, and with some of the new equipment that was just bought by the Stem Cell Institute, we can start also looking at the electrophysiology. And the hope is that we can use all of these to find treatments, pharmacological treatments, or other pathways to look at helping these people. So the negative people, the people who are negative for a mutation are all happy, but how do we help the ones that were positive for a mutation? And we'd like to end our presentation with, if you will, a call to action.
So we have summarized results, mentioning about the burden of heart disease and how we have used our research labs, combination genetics and biomedical engineering to get at this, new research tool platform. But still, as you could see, there's a lot of work to do. Okay, so we, as Anya mentioned, a call to action, so then we can continue to do research on these types of families and also understand things like aging and some of the more common types of heart disease as well.
Yeah, and for those of you who are high school students, there's plenty of things to be discovered. So come and work and explore stem cells. And this is, and as was mentioned in the beginning, this type of work, the more we fund it, the more we not only understand these mutations, but this particular mutation has parallels to aging. So plenty of people get heart disease as they age. So the more we understand the mechanisms of this, the more we understand the general mechanism of the disease.
All right, so thank you very much. Thank you for your attention.
So I think that's one of the best presentations we've had. So I laud you guys. That was just really enjoyable. I hope you guys all enjoyed it as much as I did. So, so with your patience, we have time for questions from the audience. I can run a microphone back and forth if you guys stay up here. So questions.
I can take it? Awesome. Okay. Is testing positive for the lamin AC mutation, does that mean there's a 100% chance of a of heart disease?
That's a very good question. So for in genetics, there's something called penetrance. So that's if you have a mutation, what's the chance over your lifetime that you will actually develop the disease, okay? So like I had mentioned, there's a lot of different factors. For this particular disease, unfortunately, the penetrance is very, very high. It's estimated more than 90% over the lifetime. So if someone's 40 years old and has a mutation and doesn't have disease, it doesn't really clear them. So they get in that situation of whether or not they should get Well, they definitely have to get annual cardiac evaluations, ultrasounds, EKGs, but the question is whether or not they need to put one of those intracardiac defibrillators, because they could have an arrhythmia and have sudden death. So unfortunately, the penetrance is very high, but you can also see within the families a lot of variation. Some individuals, patients present very young, and some actually, one of the patients is in her 70s, has an ICD, has dilated cardiomyopathy, but is managed medically with their cardiology. So you could see there is this one particular highly penetrant, very severe mutation. However, there are other factors that could modulate that. So it is not 100%, but then for this disorder, and some disorders are like this, where the penetrance is very high, where other disorders are very low.
Are you to the point yet where you're using the lab stem cells injected into a heart to regenerate a weak heart or a weak structure? Are you to that point yet?
So we're not to that point yet, and I'm not sure. So there are current clinical trials where people have tried to put stem cells inside hearts with the idea that they would then differentiate into cardiomyocytes, etc. And it looks, so the clinical study results that I've seen, it looks like it's safe, but it also looks like it's not effective. So there's a different type of technology that's being worked on in parallel, where you take the adult cells and you actually force them to proliferate. So they're sort of, the cells that are already in the heart sort of take a step back, and then, so that might actually win the race in terms of solving once some of the heart cells are dead, putting in something and healing the heart. I think the power of what we're doing is being able to find the mechanisms and being able to find a different way to go in there and help prevent the heart disease for a lot of these families that right now basically have something hanging over their head because they have a mutation.
Yes, I was just wondering, of the ones that did test positive for the mutation, have any of them elected to have the ICD put in?
Yes, many of them did. And that's a very difficult choice, because those pedigrees were the ones we published. And then if you actually see the number of people who were tested, there's many individuals that I didn't put on there for confidentiality. So when we offer testing, it's confidential, and then when they get the results, so these individuals have ranged from 18 years of age. So we decided, because of the ethical considerations, not to offer testing to children. There is some debate about that, but we had to make a decision there. And then if they're 18 and was at risk because of the family history, and they wanted, and we explained the benefits and the ramifications of a positive versus a negative, and they wanted to do that, we offered testing. And some individuals did, once they get positive, they would certainly have a full detailed cardiac workup, and many of those had normal cardiac function. Some had dysfunction. We actually had one gentleman whose father was affected, was asymptomatic, had testing, had a normal heart evaluation at the time. Within a year, had heart failure, had what they call a ventricular assist device until he got a heart transplant. Okay, so that was one of the youngest patients. I think he was like 29 at the time. 28 tested, 29 heart failure, very severe, heart transplant. Where we have other individuals who are 18, 19, 20, tested positive, but perhaps their cousin died. Okay, so they have to go through that. Many of the families speak with me. I give them what's current about the literature. They speak with their cardiologist, their family, and then make that personal decision. Because on the other hand, it could be life-saving, but having a device implanted also has some risk. Okay, so it's a very difficult so we could identify patients, and again, with those positive, we have to understand the disorder using an in vitro model, try to intervene, understand what factors cause some individuals to be more severe, and hopefully within the dish, screen molecules for drugs to prevent and bring it back to the patient. Okay.
When you notice that there's a problem from the implanted sheets of heart muscle, what do you do to fix it? Like, do you have to remove that sheet and then implant a different one, or can you fix it without having to do surgery?
So the sheets of muscle that you see here are actually not inside a body. They're in the dish in the lab, which allows us to demonstrate this within maybe just a month or a month and a half, while, as Mike was pointing out, it takes at least a couple of decades before you see any symptoms in the patient. So this allows us to speed up the process, and if we're looking for a cure, if we're looking for a way to do something, it's much faster for us to intervene and try it. So if, for example, we decide, and there's some indications that this is true, that the reason that these patients are having trouble is that their cells are not producing enough lamin, because one of the genes is defective. So if that is the case, if we can figure out a way to deliver lamin to these cells, then maybe we can test it really rapidly in the lab, and then if it's working in the lab, we have a pathway to go into animals, and then a pathway to go into these patients and actually look for cures.
I was going to ask a question about the movie, but it's just disappeared.
Oh.
So in that beating flap system, can you do it in enough scale that you could envisage testing thousands, perhaps tens of thousands of drugs to find drugs that might improve the beating efficiency of particular tissue that gets around the problem of putting stem cells in or doing genetic modification? And the second part of my question is it true that red wine is good for the heart? And
can you test that in a dish?
If so, how much should I drink a night?
So I can handle the first part, but I don't actually drink wine, so I hope it's not, because then I'm not doing something good. But so the first part of your question is that, as is, this is designed for laboratory use because we tweak it when we work with stem cells, etc. There are versions of this flapping system that have embedded materials in it that directly report the amount of bending, etc. So if you want to get it into a situation where it's high throughput enough for tens of thousands of drugs, you want to change the system. There's actually, so this is patented, and it was licensed recently by a company that is planning to do exactly what you're saying, is tweak this platform so that it's automated enough to sell it to pharmacological companies.
Actually, for the first one, I'd like to add, we didn't get to go in the time in the summary where you showed there was a new equipment here at the center that was just purchased, and to complement the heart and neck chip, that's where we can look at the electrical impulse. It's almost like an EKG in the dish, and then you could really rev that up as high throughput, and that's really designed for biotechnology and drug companies for drug screening. So we plan, as a complement with the heart and neck chips, to use this new multi-electrode array on our cells to screen thousands of molecules so we make a difference in the beating and the pacing. To answer your second question, my question is, how much red wine have you already had? Okay. Now, I do want to leave you with, heart disease is quite severe, many different causes. The family we did present is a very severe, case. So there are things in general, diet, exercise, and including in moderation red wine. Okay.
Thank you. That was a really terrific presentation. So you know the mutation. Why not use a gene editing system to go fix it?
Absolutely. We have built our tool, our platform, and then now we're getting to that position, high throughput, to intervene. So many different ideas which we haven't gone through. And once people have made these in vitro models, whether it's drug screening or genetic editing, to actually correct it. As we're going here, certainly in our approaches for therapies prevention as well, why don't we just go in and fix it in the dish so that we could study it very carefully and then eventually move that to an animal model. And then, as certainly for clinical trials in the near future, as some of these gene editing techniques are starting to move there. But I think our advantage is we have such a wonderful platform that we can control on multiple individuals and then move it in a stepwise appropriate manner into animals and eventually to humans.
Such an engaging talk. Thank you for that. I was wondering if you can comment on the capacity of the heart or the endogenous stem cells within the heart to regenerate, especially in the context of the retractions of Ann Versus papers last year. And is there a population of heart stem cells that are dormant that can be reactivated at all?
So from everything I know, the population of stem cells within the heart, so there's evidence that it exists, but it is so small in the human heart that we cannot rely on us being able to somehow activate it sufficiently to actually get heart regeneration. Now, there are species where you actually get heart regeneration. So in zebrafish, you can cut off about a third of the heart and it will just grow back. And there's been attempts to try, so there's a mouse strain where they mess with it sufficiently that it will also regenerate. There are some issues with it. So there's hope that maybe we can learn to somehow maintain that. But again, I think our best bet, if we're talking about regenerating in situ while the heart is still in there, is getting the cells because you have plenty of fibroblasts in there. So if we learn to manipulate the myofibroblasts, or if we learn to manipulate the heart cells that are existing to step back, proliferate, and then go back to being true heart cells, that, in terms of where the science is, I think is going to get there faster than trying to manipulate the stem cells that are there.
How did you decide to pick this particular form of cardiac disease? And how long did it take you to figure out, how to put that glue on the platform and where it comes from and all that? I mean, how long has all of this taken from when you first started, okay, we're going down this road, to where you are now?
So I'll answer the second part of the question and then give it to Mike for the first part. So developing these tools is a decade effort. So the decades. So the micropatterning, the stamping that I showed was actually originally developed a pretty long time ago for other cell types. So it wasn't specifically developed just so that we could study these families. The heart on the chip is something that I worked on when I was a postdoc. So that would have been about 10 years ago. And part of that, there was technology already there. So the masculine thin films was something that another postdoc came up with. And then I took that technology and figured out a way to have many of them and to create a chip out of it that we could realistically do studies like the one that Mike and I did. So all of this, it is not a single lab effort, it is not a single person effort, and it takes years of dedication to get there. And then the exact heart disease.
And I trained in human genetics and research as a graduate student, and then medical genetics as a clinician. So the question has remained ever since is why are certain diseases inherited? And then during this time, as you know, with the Genome Project, sequencing the gene, finding this gene for this disorder, that's what I was raised into. And then with the technology that I didn't mention, high throughput sequencing, back in the day, we could sequence one gene. Now it's actually very relatively cheap to sequence the entire genome. So with that kind of revolution in bioinformatics, then what I started to do when I was here at UCI was to do a research project to recruit families with inherited heart disease. And we recruited many different families, and one of the largest families we recruited was the first family in 2008. And then from the collection, we had other families, and it turned out that they had this same gene mutated. And when we met and combined our forces in terms of answering the same question, why is this happening, and studying it in the lab in a very careful manner, and eventually developing treatments, then it really took off. But I would agree that this is actually many, many years of effort. We've been collaborating for about 5 years on this particular study, and I'm just grateful that, and we're both grateful to share this with you. Okay, so.
I think that's probably a great place to close. Thanks everyone for coming tonight. I'm sure these guys may stick around for a couple of minutes, and let's give them a hand.